Welcome to Life Science Solutions, the podcast presented by Kymanox, where we explore the challenges, innovations, and future of our industry. In this episode, host Chris Adkins is joined by Gerry Farrell, a seasoned expert in the CDMO space with over 2 decades of experience, including leadership roles at Fujifilm Diosynth Biotechnology. Now, as an executive advisor at Kymanox, Gerry has seen the evolution of bioprocessing firsthand, from monoclonal antibodies to the complexities of gene therapy. To start, Chris is wondering about the general understanding of the manufacturing process in gene therapy and where early-stage companies often struggle. Here's Chris.
You guys obviously worked a lot in the gene therapy space whenever you were down in College Station. What do you see in terms of the products and the processes that were coming through from the various sponsor companies? Where were they at from kind of a process understanding, process characterization perspective? What was your kind of broad base observation for those folks?
They ran the whole gambit. I think a lot of the early stage companies, they have little characterization of the product or of the manufacturing process, and they just believe that the product works and want to get into patients ASAP. A lot of times, they don't really have the funds to gather that information. So, a lot of the process we saw, were, coming out of academia. Some of the larger companies more well defined, but very very basic processes, and not some of the big challenge we saw was on the analytics. Not the companies not being, as I said, don't have the funds to gather all of that initial data, so they end up with a mismatch of unreliable assays that don't tell them everything, and then with that amount of assays, it creates a burden for them and for the CDMO. Like the analytics are the most critical part of the process. Having that data, and a lot of people underestimate the time it takes to get those methods in place.
Yeah.
I think like for us, like initially, what basically just need methods that are fit for purpose, but need to be robust enough that can be transferred to CDMOs.
Yeah. And it's tough because it doesn't feel like your analytical development directly, it doesn't directly correlate to outputs of, grams of protein or loaded capsids or whatever it might be. But it has such an impact on the understanding and the characterization of your manufacturing process, right?
Yeah, it does. I think and early on, it's important that you identify and document those CQAs, or what the quality attributes of your product are, and what methods are going to help you manage those through the process. And initially for many companies, this could just be potency and titer and safety. I think what happens is people would just put a whole just shotgun approach and generate a whole load of methods that generate conflicting information and not really adding a lot of value, and then adding to the timeline. Because so to do analytics, you have to have products, so the analytics are always lagging behind the process. The more methods you have, the more mature you need to develop these methods and put them in place. I think so. It's important just to be follow your process, and as as the quality evolves, as the technology evolves, and you get a better view to where you stand with your methods and what they're telling you. Like you said initially, for us, too many methods are worse than too little, in a sense, because it's just the churn that it creates and the time that it adds to your process.
That's really interesting on the analytical side. I'm curious. I mean, obviously you've been in the space long enough to be before gene therapy and even back to some of the earlier days of monoclonal antibody, right? Not the very, very beginning, but do you see a parallel with where gene therapy is in terms of kind of the average development program that you see versus maybe what we saw for mAb processing 30 years ago? Or do you think it more just has to do with really kind of tight funding versus maybe a little bit more availability to do some of that requisite development work?
I think the challenge with gene therapy at the moment is, and just from attending the Advanced Therapies conference back in January, is that there's the process are still being defined. Today there's 5 or 6 different flavors of CAR T. There's what are the best capsids? What are the best self sorting devices? What tools are you going after? What expression systems are you using? I think with the antibodies, not that the antibodies now would be considered a simple process, but back 20 years ago it was more straightforward. You had your DG44 cell line, and you had a couple of different variations on that. But your target was, well, we're at 500 mgs. We need to be at a gram, and then we get 4 grams, 5 grams. So I think there's from a development point of view, it's probably going to follow the same path. But I think there's a lot more moving parts on the gene therapy side and the cell therapy side than what you had on the antibody side.
Yeah, we've really just kind of ratcheted up the complexity, right? And I tell folks it's that the easy drugs to develop all got developed, whether it's 20, 40, 50 years ago, right? And now we're left with harder and harder and smaller patient populations generally.
Correct. And the antibodies are still a growing field as well. We've seen from the amount of capacity that's been put in the ground, like Fujifilm's put $8 billion on the ground. Samsung continued to grow, and here in North Carolina, J&J and Amgen are putting big tanks in. So there's it continues to grow there.
Yeah, that's a good point that I want to touch on in a few minutes. I'm curious to kind of loop back, and as we're talking about early stage and startup companies, and we were both at Advanced Therapies Week not that long ago. There's a lot of kind of interest and growing optimism, I would say, right? We're recording this here in early 2025. We still kind of feel like we're thawing out from an investor appetite perspective. So do you think there's parallels with the need for better characterization and better development with what investors are looking for? Or do you think it's a bigger issue than maybe the specifics within the science, and it's more just on the commercial and the kind of economic realities?
I think a lot of it is the economic reality.
Mm-hmm.
There was a lot of optimism at the conference. Like, people said stuff's going to turn around, it's turning around. But you talk to individual people, they're also fighting, looking for funding. It seems that if you have something that's ready to be flipped, then you're getting the money.
Yeah.
I think still there's not much money going into early stage.
Yeah.
And I think part of, just from at the conference as well, a lot of people there were different tools, different approaches, different cell lines, different ways for manufacturing capsids. So a lot of maybe not directly going after indications, but building out those platforms. We have a platform that they can offer. So I think it's going to be interesting over the next couple of years how those platforms develop, and which of those move forward. I think one thing I'd seen at that conference as well was I think it's the Gartner's hype cycle for gene therapy. So we've I guess we've gone through the peak of inflated expectation. We had that. And now we've just coming out of the trough of disillusionment.
Yeah.
On the slope of enlightenment, heading towards the plateau of productivity. I think that was one of the presidents. I don't know. I should probably give credit to the person that put that together, but I'm not sure who they were. I just remember it stuck in my mind that
Yeah.
5-10 years ago it was, yes, it's great. Then we had COVID, and we just everything dropped away. Now I think it's starting to come back.
Yeah. And we have products on the market. We have successfully commercialized assets. There's a good number of them out there, and we're seeing life change that's happening from them, right? And especially they make incredible stories, right? It's much more convincing to put forth a curative story than maybe a vaccination based one, right? Where it's your success
Right.
is someone not getting something, or your success is controlling a chronic disease, right? Versus your success is truly curative in someone living who otherwise wouldn't, right?
Yeah, and I think there's a piece of marketing to be done around that as well. You just get the cost to go with that, and how you sell that with the payers. Like it's good stories. I think, but we've seen as well with a lot of the gene therapy manufacturers have been cutting back. Thermo Fisher have closing down plants. Rensler have closed down plants. Brazilians. And I think some of that maybe goes back to that peak of inflated expectations for a lot of people. Invested for a lot of CDMO space on the ground, and some of this is probably just a normalization of capacity to meet the true demand that's out there. It's a tough field to be in. Like we saw that, you saw, because you're treating such a small patient population, and then the process, the yields are so low from these processes still. So, till we get that stage where we've got to the, process optimization that, there's a big push towards platform. So if you have this platform, you've got the same, your same expression system, your same CAR-T processes going, after different indications, then you're going to minimize your costs,
Yeah.
Minimize your development time.
Yeah, no, that makes sense, right? So I mean, I think that's an interesting segue. You commented on the difficulties in the CDMO space, and it's interesting because you listed off some local to kind of, let's say, the North Carolina ecosystem and some of the growth that's going on, and we've seen, I can't remember how many, I want to say it was 10 or 11 billion dollars in investment that came in last year, for various sites and expansions and things like that. Some of which is, Fuji right here in Holly Springs, and some of the other ones that you mentioned earlier. And then at the same time, we see CDMOs shuttering some capacity, right? We have seen over the last maybe half a decade here, again, just even locally, folks, shifting away from building out internal manufacturing capacity and shifting to CDMOs, because funding got tight. So what do you make of that in the current kind of assessment in the CDMO landscape, where we see some folks growing in capacity? Some of this gets into, traditional, monoclonal antibody protein manufacturing versus gene therapy or cell therapy. But what's your kind of take on why we're seeing some folks be so successful, and then some really having to reel things back in?
So I think the success has been on the antibody side.
Mm-hmm.
Like all of the capacity, all the major investment in North Carolina in the past, like that $8-10 billion that you said, has all been into large tanks to produce antibodies. So some has been the CDMO space with Fujifilm. A lot of this is two big investments by Amgen for their in-house projects, and then J&J. I think that where you're seeing the cutback has been on the gene therapy side. And I think it hasn't progressed as quickly as people expected it to progress. Then there was an excessive capacity put on the ground for gene therapy space, and then somewhat as well as it's hard to make money at the gene therapy. So, like the Thermo and Resilience are probably doing an ROI on what they're doing and what they can make on the gene therapy compared to the antibody. You can turn a batch of antibodies every 4 days and generate tons of material.
Yeah.
And it's the same thing. Whereas the challenge of working on the gene therapy side, I think is still a tough challenge.
Mm-hmm. 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 Yeah, and it's also difficult because it's interesting. Even on the antibody side, we see so much commercial scale capacity is being built, right? I mean, CDMO and on the small company side, right? And we see some on the development side, but we certainly see more of a splash on the commercial side of things, right?
We do, and also on the antibody side, the process intensification. There's companies out there offering continuous manufacturing for antibodies. And the advantage of that, you're starting off, you got less capital investment, so you have whatever a 2K or a 4K bioreactor running continuously with a perfusion process going through continuous purification. The advantage of that, you have low capital barrier to get in, and then it's basically having the same output as those large tanks. I think one of the cool things about antibodies is going to be to see how getting the cost of goods down. Like when you get a $10 per gram antibody, which that's the stage I think antibody manufacturing is at now. It's all about cost of goods, and it's really a commodity. What's the best way to get those cost of goods down? Some are invested in the large tanks, whereas on the other side you have just Evotec have been out there with their perfusion process, continuous purification.
Mm-hmm, mm-hmm, mm-hmm. Yeah, I mean, I remember hooking up, Repligen's ATF back.
Yeah.
Oh man, I don't know, 12 years ago, right? And it was we were doing it in small, single use vessels, right? So, but really ramping up density in our N-1 reactor to then get into production, the production reactor. So, yeah, and that brings me to my next point. It's interesting how, and I cut my teeth on the single use side of the industry, right? And of course, then really went and sold single use technology and really fully drank the Kool-Aid that was the future and that was what everything was going to be in. And then, of course, everything that we talked about earlier that's being built, all enormous stainless steel tanks, and single use hasn't really gone beyond 2 or 3K, and those even still folks kind of raise their eyebrows at, right? I mean, do you have any thoughts on what that breakdown is going to continue to look like? Do you think we've kind of settled into what it's going to be, or I don't know? Everyone who's an operations person has an opinion about single use versus stainless. So what's yours? See, we'll let you offend some people.
So my personal opinion, stainless steel just scares me. I mean, you would walk into those large stainless steel plants, and just the scale and the cost, and one gasket goes awry, and you drop a whole $10 million batch.
Yeah.
That's
it's more about the volume of production at any one go, right?
Yeah, that's just personal opinion. Yeah, so I was like whatever, 15, 20 years ago, we set that what our single-use platform was going to be at Fujifilm. In my mind, stainless steel was a thing of the past.
Yeah.
I think as antibodies become prevalent, and it's growing, it's out there, the demand is out there. These drugs have taken off. So the blockbuster drugs, the biosimilars. So it's the scale. You need the scale for the cost of goods. I did think that we would have gone more towards perfusion, because of the capital investment, like the amount of money, like the billions and billions of dollars going into these stainless steel plants. So that the return on investment there is like a long time.
Yeah. And I mean, especially from a, it's one thing from a product sponsor perspective, where you're going to have the market beholden to you for your product for X number of years, right, based on exclusivity. To me, it feels much more difficult from a CDMO side, where you're reliant on clients to sign up for this really large commercial scale, right?
Correct, but I think it's a different sales process for the CDMOs with the large tanks on the ground. Then you're talking about partnerships with large pharma. So you get in with these large pharma companies. You have a view to their pipeline. You see it coming. So that's like you're not selling 20K bioreactors 4 or 5 every day of the week. These are long-term partnerships.
They're long-term deals, right?
So and you're not turning. You're maybe doing 2 or 3 products a year. So like you're not turning.
Yeah. Which makes it a lot easier operationally, right? As opposed to being in development and running, 20.
Because as the changeover cost of that scale is just tremendous. So, but again, that's large companies putting those tanks on the ground. It's about partnerships. Yeah. And they have a view to those partnerships and those deals with the large biopharma companies.
But I mean, it would make me nervous enough to build a facility for my own product that was in phase 3, right? let alone have a CDMO build it for a client's product that's in phase 3, right? Because not all phase 3 trials succeed, right? There's an example here locally of a CDMO that built out a facility for one client, and then the project failed, right? So you can have some pretty unfortunate, but that's the nature of taking the risk and maximizing your exclusivity window. So.
Yeah, and there's a lot of factors. You have the CDMOs have their models. You factor, okay, I've got one that's signed and sealed. There's a chance. You need you need a bench of products sitting behind that's going to fail, because as we know, something's going to go wrong with one or two of them.
Yeah. But it is interesting, that we never, to your point around perfusion, right? We still have not. On the antibody side, we haven't gotten to the point that we've said, okay, let's bring this back down from a sheer volume perspective, right? And work the perfusion angle more and the yield angle more, to try to. But some of it, I guess, is the regulatory inertia, right? We have a process that works, and let's keep doing it.
Yeah, you have a process that works, and you have the large companies, because again, it's the volume of material you're going to need as well. Like it's got to be. You're looking for tons of material a year. But you're right. I think it's the regulatory inertia. I think a lot of it is these large companies have their platform that all the products fit in this platform, that they're going to run with it. It works.
Because then they have all the infrastructure for it, right? They have their pilot lab, they have their scale ups, and then they're gonna make everyone fit the mold.
Which they do, and then you can see it outsourced in the CDMOs, but then you have the likes of Amgen and J&J putting extra tanks on the ground. So it's sort of, if you're going to put new stuff on the ground, why not change the process? Go to something that's got less of a capital barrier. But I don't know. It's been, I've gone back and forth for that for years. It makes, for me, single use makes the most sense. That's what I grew up with. I was not that young. We started off in stainless steel. But again, if you want 20 kegs, 30 kegs of material every week or every month, then it's the big tanks make the sense if you have the funds to build them and manufacture.
Yeah. So you've been there. You've seen that kind of let's say the single use renaissance, maybe, right? And obviously the perfusion technologies. Are there some current technologies or solutions that you're seeing out there that you're keeping your eye on and you think are gonna make a big splash in the space?
Like there's just been the continuous purification. Mm-hmm. Where you have the various multiple columns, just continuous purification of the perfusion bioreactors. Interested to see how that plays out, and if it does get a foothold. Because I think with that's going to bring down the cost of goods.
Yeah.
You have low operating costs, high yield. But yeah, on the antibody side, that's about it. The gene therapy is that's the more exciting side, right? I think.
Sure.
Where that's going to go, where you start bringing AI and machine learning start coming into those processes.
Yeah.
Like we used to have a colleague of mine from years ago. You used to talk about dry science. I'm not sure, if it was a pun intended or not, but it was, his comment was, why are scientists turning bioreactors and running experiments? That they should be, sitting at their desk, building models, running programs, analyzing data. So, I think that's where we're going to get to. When you see, you do all of your PD as the models are built out and as the data is built and the data fed back. You build out these models and you develop your process on the computer, and then just hand it off to your tech transfer group.
Yeah.
And then next step is, do you get to dry manufacturing?
Well, how far, and you're starting to waver into the digital twin idea and things like that, right? And
but getting to a stage where you take your engineering runs, you take your shakedown runs out of the picture, with the data feedback that you have all that stuff because I'm no expert on AI, but just reading
as two operations guys, I'm sure this seems terrible. It's terrifying to us, right? I can't imagine not having that engineering activity, and I can still tell you how many deviations we had on our first GMP run, right? And we did engineering runs, so
right. But that's, and maybe you're not full to that, but using this machine learning, this data feedback to prevent or predict deviations. As you're building data, as you're building models.
Yeah. There's a tremendous amount of work on the discovery side, right?
Yeah.
And, for molecule discovery and things like that, but we're still not. I don't know how much they improve the percentages. I'm sure there's some marketing material out there for some drug discovery AI ML companies that would tell you, but it's still a challenge to simulate a full biological system, whether it's the human body or it's the process, right?
Yeah, of course it is, but that's what we're getting to. As you build more information, and then if you get the scientists, the manufacturing guys, the clinicians together, and even the patients, and you start building those models, all of that data is fed back. How far can you go with reduced clinical trials or reduced prep time, reduced manufacturing? Because today it's the cost of goods on the gene therapy side is what's killing everybody.
Yeah.
So how you get those cost of goods down? One is the platform processes.
Yep.
And how you manage that. And platforms go back and forth. They're in again, they're out again. But I think it is once. Today there's so many different versions of platforms out there. What's the best platform? What's the DG44 of the gene therapy side?
What's gonna ultimately win, right? We're still in a phase where the race is still actively being run, right? So, no, I think that's good. So, anything else?
I think one thing back to the analytics. I think a lot of customers or sponsors, don't take advantage of the CDMO. They tend to just dump the package on them. You go to CDMO because they have that level of experience. So, to take advantage of that when you're developing your analytical package and what you need for your process.
Mm-hmm.
Because
involve them earlier and
like us, because the CDMO has shared ownership in the process, like reputable CDMOs. Well, I'm sure there's. I don't mean to say there's non-reputable CDMOs out there, but just you find a lot of time you hear, like, from the sponsor side all this presentation about partnership and quality agreements, and all that stuff's critically important and you need it. But there's also a piece that you need to leverage experience. You're paying for that experience, so leverage that experience with the CDMO, and when they give input, take it, because they've been down the path. They've seen the products go through. They've seen the regulatory approaches and what's needed.
Yeah. If you're having to tell them exactly what to do all the time, we would say you probably haven't chosen the right CDMO.
Yeah, correct.
Right?
Yeah.
You've either got them over their skis or something else worse, right? So I would be inclined to agree. So, well, excellent. Well, Gerry, really appreciate the time. Thanks for joining us on the podcast today, and look forward to future conversations.
Thank you, Chris. Enjoyed it.
Thank you for tuning in to Life Science Solutions presented by Kymanox. A big thank you to today's guest, Gerry Farrell, for sharing his insights on the evolving CDMO landscape, the challenges of gene therapy manufacturing, and the future of bioprocessing. For more information on Kymanox and how we help bring life science products to market faster and more efficiently, visit Kymanox.com. This episode was edited and produced by Walk West. Until next time, this is Life Science Solutions, where challenge meets innovation.